You're already familiar with the process of mitosis, so now you need to learn the process of meiosis. Meiosis is the creation of gametes by cell division. It has a lot in common with mitosis, but there are also some pretty important differences, so the best place to start is with a comparison and contrast between the two processes. Mitosis results in two daughter cells that are identical genetically. It's important that they're genetically identical because they result in body cells or somatic cells. Those are cells that it's very important that they're identical to the cell that they started as or originated as, because they already are differentiated. So they already have a function and they need to maintain that function. Mathematically, we say that mitosis results in diploid cells, which we call 2n. So for a human, we would say 2n equals 46. And this is called diploid. Diploid simply means a cell that possesses a complete complement of chromosomes, one originating from the biological mother, one originating from the biological father. So, for example, you have 23 chromosomes from your mother, 23 from your father, leading to a diploid number of 46. All cells in mitosis will result in the diploid number of chromosomes. Mitosis has one instance of interphase, and also only one cell division. Meiosis, on the other hand, results in 4 genetically unique cells. These are not body cells or somatic cells, but instead these are gametes, so sex cells. Instead of being diploid, instead these are haploid, so we represent that with the equation n. So for a human being, n equals 23, and we call that haploid. And that simply means you have half of the number of chromosomes that the normal body cell would have. In meiosis, there's one interphase again, so that's just like mitosis, but 2 cell divisions. That's part of what gives meiosis the name reduction division, because it reduces the number of chromosomes because it only replicates DNA once but divides twice. These are the major differences between mitosis and meiosis. There'll be some other subtle things along the way, but they're going to include a lot of the very familiar words we already know. This is an overview of what happens during the process of meiosis. So here in picture A, you'll notice that we're in interphase before synthesis. We know it's before synthesis because there are only 2 chromosomes here, 2 chromatids. You can see them. There's one, there's the other. Those chromosomes duplicate, so we now still have 2 because they're attached by their centromere, so they still only count as 2 chromosomes. But they have 2 chromatids joined together. After the first round of meiosis, so right in this area, right here, is where meiosis 1 takes place, the first division. And you'll notice that it reduces the number of chromosomes in the daughter cell. We went from 2n equals 2, because here's 1 chromosome there, and here's the other chromosome there, so 1, 2, to n equals 1. There's only 1 chromosome in each cell. This occasionally confuses students why we consider only 1 chromosome when we have 2 sister chromatids joined together. But because they're joined together by the centromere, they're still considered only 1 chromosome. After meiosis 2, which occurs between C and D, you'll notice we've reduced to still n equals 1, but now there's only a single chromosome, not a duplicated chromosome, in each cell. So that's the resulting product. We have 2 divisions where we end up with 4 haploid cells. Meiosis 1 is called reduction division, specifically referred to meiosis 1 as reduction division, because that's where we go from 2n to n. So we go from diploid to haploid. So we consider that reduction division because it reduces the number of chromosomes. Looking at our first picture here, we're looking at everything that occurs during the first phase of meiosis, which we name very similarly to the first phase of mitosis. We call this prophase, and these are all of the events that occur during prophase. But here we'll call it prophase 1, which is the first phase of meiosis 1. During prophase 1, you'll notice that we're going to have a nuclear envelope dissolve. So you'll see what was peach in the left diagram disappears in the center in the end diagram. Centrioles are going to migrate to opposite sides of the cell. Our chromosomes condense to become visible. And spindle fibers form. Now this is all really, truthfully, absolutely identical to prophase of mitosis, except something strange happens here that I want you to pay close attention to. We see our chromosomes in the first diagram here. And in that first diagram, there are 2 similarly sized chromosomes and 2 other similarly sized chromosomes. In prophase, as we migrate through prophase, you'll notice that the similarly sized chromosomes are going to line up with each other. And then in the last diagram, they've lined up completely, and you'll notice down at the bottom, they've like switched arms. It's like the end of the purple one came off and the end of the green one came off, and they switched places. These chromosomes have special names. The ones of similar size are called homologous chromosomes. And those homologous chromosomes are going to pair during prophase 1. Homologous chromosomes simply refer to chromosomes that contain the same genetic information. So here we have the purple one that we'll say is information from mom, and the green one that is the same genes, but they were donated or provided by dad. So they're the 2 chromosomes that belong together, that contain all of the same genetic information, but originated from different parents. And that's what's shown here is one homologous pair, that's what we call them, and here's the other homologous pair. In prophase 1, the homologs will align next to each other, and they cross over. So their arms are going to link. We call that crossing over. And just like we see in that that end diagram above, you'll notice that those ends like switched places. We said that the green and purple kind of broke off and switched arms. We call that synapsis. And it's not a problem that they switched places like that, because all of the genes are located on the chromosomes in the exact same spots. So if they're located in the same spot and you switched arms or hands, think of it that way, like when you were a little kid playing Red Rover, when you had arms linked together. If when you tugged on your partner's arm trying to make sure that your chain stayed tight, if your forearm and their forearm switched, while it would have been awkward and unusual and thank goodness can't happen, had it happened, it wouldn't really have been a big deal because you would have exchanged a forearm for a forearm. Same thing here, you exchange one copy of a gene for another copy of the gene, but they're the same gene, so it's not a big deal. Actually, it's very beneficial during prophase 1. So what I've underlined on this slide are the differences between prophase of mitosis and prophase 1 of meiosis. The things that aren't underlined are all similarities, but the things that are underlined are differences. Here you should recognize this as metaphase, because in metaphase, you'll remember chromosomes align at the metaphase plate. So at the metaphase plate, you'll notice this is slightly different than in mitosis, where mitosis they would have aligned horizontally across the metaphase plate where all 4 chromosomes would have been spread out. Here, they align as homologs. So I'll underline that again because that one is another difference between metaphase and metaphase 1, where we would have seen, and let's see, in mitosis, they would have all aligned that way across the center, so across our equator, all lined up separately. But since this is, in fact, meiosis, specifically metaphase of meiosis, metaphase 1, you'll notice that our homologs are still stacked on top of each other and not aligned separately. That's unique to metaphase 1. Then we come to anaphase 1. In anaphase 1, chromosomes are going to be pulled apart, so chromosomes migrate to opposite poles. Except the difference here is the centromere does not dissolve. So instead of having sister chromatids being pulled to opposite sides of the cell, here it's whole chromosomes that are being pulled to opposite sides of the cell. And you can more clearly see the crossing over in synapsis that had occurred during prophase 1 as we examine these arms on those 2 different chromosomes. That's essentially all that's going to happen in anaphase 1, and really, truthfully, that's the only difference, is that the centromeres don't dissolve. So then we come to telophase and cytokinesis. So this is telophase 1 and cytokinesis. We don't label it cytokinesis 1, we just call it cytokinesis. Here we have our chromosomes that are going to be pulled or have finished migrating all the way to opposite sides. The new nuclear envelope forms. And cytokinesis pinches the contents, the cytoplasmic materials, pinches it in half to form 2 different cells. This really, truthfully, isn't any different than telophase of mitosis. You'll have 2 nuclei resulting. The only change here for us is going to be the amount of DNA in each of those cells. If you look at it, here we started in that first slide of prophase 1 as 2n equals 4, and we're ending at n equals 2. So we've already reduced the number of chromosomes by half simply through 1 division. And that's all because our centromeres didn't dissolve, but rather stayed together. So one of the differences then is going to be that the cells are now haploid. So each nucleus contains half the amount of DNA that the parent cell had. Meiosis 2 is going to look a lot more similar to mitosis. Most of the things that were different during meiosis 1 are now actually the same as mitosis during meiosis 2. Here we have the start of meiosis 2 with prophase 2. All of our events from prophase we recognize: nuclear envelope dissolves, centrioles migrate to opposite poles of the cell, spindle fibers form, and chromosomes actually remain condensed. Our chromatin remains condensed into chromosomes. Then we have metaphase 2. And in metaphase 2, the chromosomes align at the metaphase plate. Here, they're not aligned as homologs because there are no longer homologs. They've been separated into separate cells. They're just aligned along the equator as they would in mitosis, lined up along the equator horizontally. During anaphase 2, the centromere does dissolve here. So this time, when they get pulled apart, the sister chromatids do separate, which is unique from meiosis 1, but is actually the same as mitosis. And then we have telophase 2 and cytokinesis again. Where we have the two new nuclear envelopes forming. Here they're shown in peach. And then cytokinesis, we're going to have the furrow form again, so it can be pinched into 4 individual cells. Here, we started meiosis 2 as N, and we end as N, all because of the dissolving of that centromere. That's the end of meiosis, essentially. Something that might be helpful for you is if you go back and take a look at these diagrams, count the number of chromosomes so you can see where we start as 2N, we reduce after meiosis 1 to N, and we remain at N through meiosis 2.